the chat. There we go. Okay, cool. So I'm going to talk to you guys a bit about my research, which is on CTCF chromatin dynamics and leukemia. So I'm going to try to keep this as Zoom interactive as possible because I've been working on this for too long and it's kind of like a niche within biology. So I'm going to try to give as much background as I possibly can. But if at any point you guys are confused or have questions or want me to explain something more, please just jump in and I'll go ahead and explain that for you so that we're all on the same page and not super, super bored. Or you just don't like biochemistry, which is fine too. So I'm going to start off with, first by going to the next slide. Okay, so let me start off by giving an overview of gene expression. So in our cells, all of our cells have DNA, and this DNA codes for genes. So in order for a gene to be expressed into a functional product, it undergoes this process known as transcription, which kind of creates a copy of the gene into this thing called messenger RNA, which is kind of just another version of the same message that was coded within DNA. And this here is a picture. This here is a picture of how this process of transcription might happen. So you have some components here that can take a template strand of DNA and transcribe it into this string of messenger RNA. And then this messenger RNA can then be translated into a component known as a protein. And the protein is the actual functional unit that does everything in the cell. So is this familiar to everyone, or was that too fast? Okay, the chat looks pretty empty, so I'm assuming that everyone is pretty familiar with this, hopefully. Okay, so I'm going to start off with a question for you guys, because we all know, or now you know, that all the cells in your body share the same DNA that codes for the same genes, more or less. So how do we get different cell types? Because I have cells that make skin, that form skin, I have cells that form liver, I have cells that form heart. So they all have the same DNA. How is it that they're different? Any ideas? Okay. It's still a little silent. So let me give magic. Yeah, basically. So think of it, the problem as your cell is a factory and all the proteins are production outputs. So for example, you have this factory that produces ice cream sandwiches in this picture, okay? And you have all these little processes, some machine is baking the cookies, some machine is making the ice cream, some machine is making the plastic wrapper. So you have all these instructions out there. How do you differentiate those or control those instructions such that you get a nice ice cream sandwich instead of a splodge of ice cream and cookies? This is a very general question. So say you have the machine that helps these cookies slide down into this little slot here. What can you do with that part of the machine? You can turn it on and off, you can change the rate, you can change the temperature, presumably. I also lost my mouse, so give me one sec. Okay. So does anyone have any ideas as to how we might get different cell types and different products from the same set of instructions? Okay, so what I'm trying to get at here is that even if you have the same starting code for every single cell, it doesn't mean every cell uses the code in the same way. And the way to do that is by regulating gene expression. And on a super high level, you can think of like, for every given gene, a cell can make a decision as to whether or not that gene is on or off. So is it expressed or not expressed? If it is it expressed quickly? Like, what's the rate of expression? Like, do you need a lot of this product, or do you need not so much and like at a much slower rate? And once you know whether or not you want to turn that gene on or off, or the rate at which you're producing it, you might also want to change like the type of the gene, and that can be done through some of these downstream regulatory processes here. So the focus of my research is very much on transcriptional control, or even a higher level of transcriptional control. So I'm going to talk a little bit more about that. Does anyone have any questions so far? Or is this clear? Okay, I guess it's clear. So I talked a bit about how genes can be turned on and off, and the rate of their expression can be varied. So one way in which cells can do this is by regulatory elements. So these are actually parts of DNA that don't actually code for anything. So like if you transcribe it, they don't form a protein. But what they do code for is an instruction to whatever target they have as to whether to transcribe faster, transcribe slower, or not transcribe at all. And these elements, or a very common set of these non-coding elements, are known as enhancers. And the way that these enhancers work is that they need to be physically close to a gene. And so in this diagram, which is like a little overly complicated, but I just want you to focus on the gene body portion and the core promoter. So the promoter is just like the beginning part of the gene that is where the gene starts to be transcribed. And when you have a non-coding element or regulatory element, such as an enhancer, come into contact with a gene, that kind of tells the cell like, okay, I want you to make this gene even faster than before, or start making loads of this gene. And sometimes, even though it's called an enhancer, if it comes into contact with this target gene, it can actually tell the cell to stop making more of that gene. So there's a lot of really high levels of regulation of your gene expression. And by all this variation, that's where you get all your different cell types. So I mentioned that these non-coding elements act through coming into physical contact with their target genes. So you can imagine that they're not just kind of like floating around in space and come into contact only when they happen to come into contact. Like this process is highly regulated into a bunch of local interactions. And when you have a bunch of these local interactions, they're actually organized into larger interaction neighborhoods that we call topological associating domains. So this is a picture of all the chromosomes in a cell. So all the little wires represent the DNA bound to proteins, which is called chromatin. And they're kind of found in these squiggles, but these are actually highly organized squiggles, even though in this picture they are not organized. And each of these blobs with a squiggle in them is representing the amalgamation of local interactions that forms this big neighborhood. So there's a lot of spatial organization to this. So how do we capture these interactions? Because right now with the current technology, it is not possible for us to see where these loops and interactions are occurring on the DNA level. Like we just don't have the resolution to do that. So a way to infer that biochemically is to take this technique called Hi-C. It is not JUICE, but it stands for like high throughput chromatin conformation capture. And the basis of this technique is that you use a chemical agent known as you use a chemical agent to cross-link the DNA, and basically what this does is it kind of freezes the cell in its current spatial state, such that any piece of DNA or any protein that is within 1 angstrom of each other is then bound by these chemical crosslinks. So you kind of froze the nucleus in this at this one point in time. So then you take this cross-linked cell with all the contacted DNA together, and then you cut it up into pieces with a protein known as the restriction enzyme, whose job is to actually chop up DNA into pieces. But it cannot chop a cross-link. So you kind of preserve all those frozen points of contact. So then once you have all these contacts that are now chopped out, you can label them. And we label them with this component called biotin. It's a biochemical component that's very useful because we can, it's very highly specifically recognized by this other component known as streptavidin. So we kind of take advantage of this very specific and strong bond between biotin and streptavidin, and use that to isolate out all these chopped up crosslinks that we've then labeled. So you can see that there's like a blue strand and an orange strand. So they represent like different sections of DNA that were brought together in close physical proximity. So to capture those, what we can do is after labeling them with biotin, we then stick them together with a DNA ligase, which is another protein whose function is to basically glue free ends of DNA together. So now you kind of have like a DNA chimera, which is like DNA along a string that is comprised of different portions of DNA that were previously unconnected. So with these like chimeras, you can then sequence the whole length of the chimera, and based on sequence alignment, you can infer where the blue piece of DNA came from and where the orange piece of DNA came from. So I'm gonna go on a little bit of a tangent here and mention, because I know some of you guys are interested in like CS and like, mostly I heard CS actually, and like engineering. So this is an example of how computer science is absolutely crucial, and other fields also, not just computer science, is absolutely crucial for this type of biology, because this whole algorithm of sequence alignment like was not figured out by a biologist by any means. Like it took a group of very brilliant like statisticians, mathematicians, and computer scientists to actually figure out the algorithm behind very specific and high fidelity sequence alignment. Do you guys know what I'm talking about when I say sequence alignment? Someone say yes or no, please. No, okay. So what I mean by sequence alignment is when we sequence a cell's DNA, we're using human cells. So in order to figure out where a piece of DNA came from, we already know what the sequence of the human genome is. A big group figured that out back in the early 2000s. So in order for us to take an unknown piece of DNA and figure out where it came from, we align it or match it to the reference human genome. And from there we can say, okay, this piece of DNA came from chromosome 1, the very tip, or something like that. Does that make sense? Okay, cool. So once we do all this sequencing, the way we can visualize these interactions is through an interaction heat map. And an example of that is shown on the right. But I'm going to give like a more detailed tutorial as to how to read this. So basically what we get is the square on the right with a bunch of red dots. And the way to read this is it's kind of like a correlation map where on the x-axis is the length of the chromosome, and on the y-axis is also the length of the chromosome. So if you take, for example, point A on a piece of DNA and point B on a piece of DNA, you can see them on the contact map just by, you know, and this is really hard for me to show now because my mouse isn't showing up, but basically from point A and point B, you can go down either the y-axis or the x-axis and see the point of interaction. So basically what this is showing you is that at the point where the arrow coming down from A and the arrow going right from B is meeting, there's like a red dot there. And the intensity of that red dot shows you how many times you captured that contact between point A and point B. So what this map is showing you is that A and B seem to interact quite a bit. So, if you were to pull out DNA on a string, A and B have their certain locations, but in actual 3D space, they're probably pretty close. So the bottom circle where it's all like kind of coiled up together is an approximation of where they're actually located in 3D space. Any questions so far? I'm gonna move on. So we have all these interactions and interaction neighborhoods, and the way that these interactions are regulated partially is by CTCF. So for every like loop that you can see based on your Hi-C assay and like all these schematics, every loop has this protein called CTCF, which is found at the base of the loop. And CTCF is also found at the boundaries of these giant interaction neighborhoods. And what it does is first it just forms like it makes up like the coordinates of the actual interaction neighborhood, and it insulates this neighborhood from interacting with other parts of the genome. So this is the protein that I study, and it's interesting to me, not only because of its basic biology function, but also because we know that it has a function in preventing tumors. So there was a study done previously where they took mice and they genetically knocked out half of the mice's normal amount of CTCF. So we call this a heterozygous knockout, and it's represented on the graph by the red line, and the notation is CTCF plus/minus, indicating that it has only one copy as opposed to two copies. And what they found was that these heterozygous knockout mice developed cancers spontaneously at a much faster rate than normal wild-type mice. So this is showing that normal levels of CTCF are actually required to prevent development of cancer. And in a study of a bunch of human ALL patients or T-cell acute lymphoblastic leukemia patients, they found that a lot of these patients had a mutation in CTCF, and this mutation actually co-occurs with a Notch1 mutation, which is shown on the bottom right, but that's just some extra information. So we know that this protein CTCF likely has some function in leukemia. So before I go further into that, let me give you some background on what T-cell acute lymphoblastic leukemia is. So a leukemia is a malignancy of blood cells, specifically white blood cells. And in this case, it's T-cell acute lymphoblastic leukemia. So it's a malignancy of T cells. And what happens is, these T cells have something wrong in their development, and instead of differentiating into normal T cells, they're kind of arrested at some like precursor early stage. And not only that, but they also proliferate uncontrollably. So these patients have huge numbers of leukemia cells, and once they aggregate, they create all sorts of problems for these patients. And on the very right, you can see these 3 test tubes. These are samples of blood that's been centrifuged or spun down at a very fast rate. And when you do that to blood, you can actually separate out its 3 major components, which are the red blood cells, the buffy coat, which contains the majority of the white blood cells, and plasma. So on the left, you can see the healthy state. So normal people don't have that many white blood cells circulating. Like, you actually don't need that much hanging around. The reactive state is, say, like you have an infection of some sort, then your body like produces a few more white blood cells to fight the infection. But you can see on the very right, like in these leukemia patients, the buffy coat with all the white blood cells is much bigger than in healthy patients or even the patients with some sort of like virus. And this is actually like a mild case. Like if you have like full-on TL patients in like the blast phase, they can be even larger than this. So it's very significant, and if untreated, it's definitely fatal quickly. So the treatment for this type of leukemia is mostly chemotherapy, and those are just drugs that are highly toxic and meant to kill the tumor cells. Other options are also radiation. In leukemia, that's mostly used for metastases. And as a last resort, there's also the option of a stem cell transplant. So in a stem cell transplant, what they do is they kind of remove all of the, not remove, but they kill off basically all of the patient's original bone marrow and try to replace it with healthy bone marrow. So, okay, so these are the 3 types of treatments that are available. And actually in leukemia, it's the rate of treatment success is pretty high for kids because kids can actually tolerate chemotherapy drugs much better than adults. However, a problem with these cancer survivors, first of all, not all patients respond well to therapy. And second, those patients who do survive their chemotherapy tend to have a much higher incidence of adverse events later on in life. And that's what the graph on the right is trying to show you. So in this study, they looked at all childhood cancer survivors, of which leukemia is actually one of the most common childhood cancers. And you can see that the incidence of some sort of disabling or fatal event is much higher in cancer survivors than in their siblings. And that's pretty much a direct consequence of how toxic chemotherapy is. So clearly these patients need some sort of like targeted therapy. And we think perturbing CTCF might be a way to get at this targeted therapy so these patients can have better lives. So one way that CTCF can be dysregulated in TAL is, so I mentioned, let me go back. Okay, so we talked about like how enhancers can only act on genes if they're in physical proximity. So a way for them to be in physical proximity with their target is if they're within a loop, right? So, and that's kind of modeled here. So on the right, you can see in a healthy cell, there's like this loop that in this example actually separates an oncogene from, or like a bad gene that you don't want to be activated from an enhancer element. So in normal cells, these 2 components are not touching each other, okay? Like you don't want the oncogene like doing stuff in your healthy cells. But if you get rid of the loop, say for some reason CTCF is no longer there and the loop no longer forms, then you have this bad gene and this enhancer element that can now have a much higher probability of contacting each other and activating the bad gene, leading to disease. And I think on the left, it's a bit complicated, but that's exactly what they found to occur in T-cell leukemia at one CTCF site that they found was deleted in a lot of leukemia patients. And it activated this bad gene that's also known as TAL1. So seeing this data that came out in 2016, I and my lab thought that, there's a lot of CTCF sites in the genome, and we think that there's probably a lot more of these types of CTCF sites hanging out there. So what we found was that there's actually a set of CTCF binding sites that are totally unique in leukemia. So they're binding to places where in any other cell type they're not found, or in a bunch of different cells, like every single cell you can think of, CTCF is bound to a certain location, but at that certain location in leukemia, it's actually lost. We think the specificity might allow for some room for targeting here. And we also found that these unique CTCF sites actually do something. So as I mentioned before, CTCF regulates looping interactions. So if we find unique sets of CTCF sites in leukemia, the logical inference to make is that these unique sites are probably changing the landscape of chromatin interactions. And that's basically exactly what we found. So this on the left is a comparative high-C contact map. This is from my data. And if you sum up all the interactions at lost CTCF sites versus constitutive or like every other CTCF site versus those sites that are gained, what this graph is trying to show you is that interactions actually do change in that at lost sites, they're much less frequent in TAL, and at gained sites, they're much more frequent in TAL. And we also found that the binding of these CTCF sites is dependent on an oncogene or a so-called bad gene in cancer. So if we inhibit this gene in T cells using a drug called gamma secretase, and the thing on the left here, yeah, my mouse disappeared again. I don't know why. This is a sign of my age that I can't get the mouse to show up. But anyway, so if you inhibit this bad gene, which is known as Notch1, and that's shown in the middle band, you can see the level of Notch1 is much more decreased compared to the first. What we found is that a lot of these uniquely gained CTCF sites disappear. And then if we recover Notch1, like you see in the third band, a lot of these unique CTCF sites come back. So from this, we inferred that you need Notch1 there in order for these unique CTCF sites to actually bind. So that's kind of where we've gotten so far. We have a lot of other data as well, but I thought maybe for an overview, it's probably better to not go over it. But if you're interested, hopefully this paper will come out soonish. But yeah, so I'll just conclude with some acknowledgements. It's a big collaborative effort between several labs here at Northwestern as well as at other schools. And yeah, it's just a flavor of some of the work that I've been doing in leukemia. Anyone have any questions? Oh, okay, cool. Stop the share.
All right. Looks like no questions. Crystal clear, Celeste.
Must have explained things exhaustively.
Cool. Well, thanks, Celeste. Let's see what else we have. So I made a brief PowerPoint. Let me share it. And Stephen and Celeste are welcome to join in whenever they want. But there was some interest previously in talking a little bit more about specialties. I know in a previous session, we talked a little bit more about different jobs that are available available in healthcare in general. And sorry, let me make this. And so now we're going to talk a little bit more about medical specialties. And so before we start, really, there's these are going to be talking about specialties mostly exclusive to students who, medical students and who have an MD or a DO degree. But you can imagine that anyone working in these specialties often has a huge team of experts with them, including nurses, physical therapists, and a lot of the other special medical, like healthcare specialists that we talked about. And who's working in what fields depends on and what is necessary for that field. So you can imagine in some of the rehabilitation specialties, there's a lot more physical and occupational therapists, and in some of the surgical specialties, you may see surgical assistants, something like that. So this can be kind of a broad overview of how the medical system, at least in the United States, is set up based into specializations. And you may have seen this in your own lives. If you have ever needed specialized care, you may have had to go to your primary care provider, who's generally a general practitioner, and they may have had to refer you to specialty care. And that's especially true if you needed surgery or other specialty care. Okay, let me see if I can find the chat real quick. I seem to have lost it. Okay, I'm too old.
That's pretty quiet.
Okay, I imagine so.
Yeah.
All right, so I'll just continue talking, and you guys can stop me if I have. Oh, there it is. Okay, so first of all, one of the important things to talk about when you're talking about medical training is what is the role of every person in the MD pipeline or DO pipeline. And so all of us are medical students. So we're all the way, this is oriented from left to right, but generally it would be talked about from bottom to top in that same order. So medical students tend to be the bottom of the, hold on, you saw that picture? Yeah, I did not draw these pictures, so you may have seen them before. But yeah, medical students tend to be at the bottom of the pyramid, meaning there are generally a lot of them, and they're generally less useful than most of the other people working. But they're learning how to do the job. An intern is a medical student who has just gotten their MD and is just starting their residency. An intern is in the first year of their residency. And then a resident is someone who is I think interns are also included in residents. The terminology can be a bit confusing, not that important. The residents are MDs who have obtained their MD from medical school and are now doing specialized training to learn how to do a certain specialty. And then once you finish your residency, which can be anywhere from 2 to 7 years long, you can actually go to further training to join a subspecialty. And those people that are doing that training are called fellows. Then once you've finished either your residency and or your fellowship, and have finished your training, then you are known as an attending, so an attending physician. And so a lot of medical students will either be supervised by residents, especially more senior residents, or by attendings. So it tends to be that one attending will supervise several fellows and residents, and then each of those people may, in a teaching hospital at least, will supervise younger medical students who are still learning. And it operates kind of like a pyramid or a Ponzi scheme in that way. All right, so this is my own breakdown of how I think about medical specialty or specialties in medicine. And I like to think of them in terms of medical specialties, surgical specialties, other, and pediatrics. So medical specialties, the residents tend to go through an internal medicine residency, which is somewhere around 3 to 5 years. And then they can further go on to do a fellowship and learn more about a specific field. Surgical specialties, these are people who are going to become surgeons, and they generally, but not always, go through a general surgery residency, which is generally 5 years. And then they can further specialize as well, or can work as a general surgeon. I'll skip to pediatrics. You guys all know what pediatricians do. They work with children or young adults. In most cases, though, in some cases, people are with congenital diseases and other things are followed by pediatricians their entire lives. And then other are residencies, which generally require a 1-year internship in medicine or surgery, and sometimes that's up to the particular person, or sometimes it's mandated based on the specialty. And then they continue on with their specialty residency, but this is not always the case. And just in general, throughout this entire presentation, there may be some metrics and things that are presented as absolutes, but there are really no absolutes here. There's a huge number of training paths that are available to medical specialists. And so it really depends on the program, but we're just trying to present a general overview here. So Oh yeah, and some of these comics, none of them should be offensive, so sorry if they are. So medical specialties, among these, and please stop us if you want to talk about any of them, or you can talk about them at the end. But people who specialize in allergy or immunology, and then some of the kind of organ system specialists, like cardiology, who specializes in the care of the heart, and circulatory system, endocrinology, who focuses on endocrine disorders, gastroenterology, focuses on the gastrointestinal tract, hematology, blood. Infectious diseases are gaining some notoriety right now. Nephrology focuses on kidneys, oncology on cancer, pulmonology on lungs, and rheumatology on rheumatological disorders. So these specialties generally require medical residency first, which is usually 3 to 4 years. And then these would be subspecialties of medicine. But there are a lot more than this. These are just some of the bigger ones. And they all require different amounts of training. So this is a busy flow chart, but this is from, I don't remember exactly where it's from because I can't see it, but in my notes, but this is from one of the training pathways, and they show kind of the huge variety of ways that people can do training in medicine. And so you can see on the top right where general internal medicine is. You can become a primary care or hospital medicine doctor, or have your own practice if you just do the residency and don't do any fellowship. And so that's pretty common to do that. And a lot of the doctors that you guys have probably seen, if they haven't done pediatrics training and they do both kids and adults, will have done just a general internal medicine residency and then started working. And this is true of some doctors in the hospital as well. And then you can see on the left where it says fellowship training, there's a huge number, and actually it even continues down to the bottom where they couldn't fit everything horizontally, so they just added at the bottom for some reason. But you can see there's a huge number of different fellowships that can be done. And then some of these fellowships, like cardiovascular disease, have even further subspecializations where you can do a second fellowship. So you could choose to do your fellowship in cardiovascular disease, and then you can do it in, then you can do another fellowship in particular if you want to study heart failure and transplant cardiology or cardiac electrophysiology or something like that. So there's quite a number of different pathways here, and medicine tends to be one of the most common large blocks that people go to after medical school. Surgical specialties, unfortunately, I was unable to find a nice chart, so I made a nice Excel table for you guys. But you guys are probably all familiar with what surgeons do. Generally, it involves cutting people open and hopefully fixing things. But there's a lot of subspecialties here. I've listed some on the left, and then you see on the table on the right, a lot of the surgical residencies, so that's the ones at the top, require a general surgery residency, and that's a 5-year often residency. Though a lot of these residencies are also starting to incorporate research, and they actually get even longer. Most of the surgical subspecialties, if you don't stay as a general surgeon, generally require another 2 years or more of fellowship. So that's 7 years after medical school of training, if not more. There are some surgical specialties which have their own residencies, but require generally 1 year or more in general surgical training. So those include like ENT has a 1-year general surgery for an intern year, and then 3 years of training, especially in otolaryngology. And you can see that this is the case for most. The exceptions are in obstetrics and gynecology or gynecologic oncology. Generally, these are approached through going through OB-GYN, so obstetrics and gynecology as your residency, and then potentially doing a fellowship afterwards. But yeah, there's a lot of exceptions. Some of these are applied to directly. So all the ones that don't require a general surgery residency, you would apply to directly out of medical school generally. And some of them have combined programs, which are shorter. For instance, vascular surgery can have a combined program that takes a total of 5 years instead of 7. And then I kind of lumped together a whole bunch of specialties. Probably not appropriate to do it this way, but to include a lot of specialties which don't fit neatly into medicine or surgery, and generally have residencies that are not approached through either one of those. So that includes things like anesthesiology. Dermatology is always a hot topic. It's known as one of the lifestyle specialties. Emergency medicine has its own residency, and then the radiology, both interventional and diagnostic, have their own residencies as well. And then pediatrics. Pediatrics is a really large field. Obviously, kids go to the doctor quite a bit and have annual checkups, and there's a lot of effort that goes in here. So pediatrics has some of its own specialties, and these can include adolescent medicine and neonatology, which are obviously only relevant to kids. Child neurology has its own residency as well. And then in addition to that, most medical and surgical specialties also have the pediatric equivalent. So you can have a pediatric surgeon or a pediatric neurosurgeon if you wanted to specialize there. And as you can imagine, the more different specializations you have, the longer the training is. So some of the very, very specialized pediatric doctors, especially the surgeons, can have more than 10 years of residency and fellowship training. So that kind of begs the question, at least for us, and this is maybe not quite as relevant to you guys yet, but something that you guys can think about now even. How to choose your specialty, or think about which specialties you might be interested in. I know that Sage has told us previously that she has a particular interest in pain medicine, but I think that the rest of us are pretty much undecided or not quite decided yet on what we want to do. But medical students have a variety of inputs to choosing their specialty. And so some people come into medical school already knowing what they want to do, and some of them change their mind, but a lot of them don't. And this can often involve following family mentors or other people that they look up to. In the case of people who are involved in research, and that's all of us, oftentimes the research interests of that individual can tie into what specialties they would consider, and this might, because they may involve their specialty in their future research. I think one of the major contributors to what specialty people choose is the clinical exposure during the 3rd and 4th years of medical school. And this is when medical students are spending a vast majority of their time in the clinic, learning about all these different specialties and how to take care of patients in a hands-on way. It's not that there isn't any clinical exposure in the first 2 years of medical school. There certainly is at most medical schools these days. But the vast majority of it occurs during the 3rd and 4th years, and you have a lot more responsibility as you get to be an older student. And there are other factors, and we can talk about a couple of them, but obviously there's a huge number of factors, and everybody has to decide on what the most important factors are for them. We'll talk about them in a second. I just wanted to briefly mention so you can see that this is Northwestern's requirements. They're a little bit different based on schools, but almost identical. And this tells what rotations are required for 3rd and 4th year students. And so you can see medicine and surgery are both required. And these are some of the longer blocks that you have to spend, I don't remember, 8 weeks or 12 weeks on medicine and surgery. I can't remember the exact number, but you spend much longer on those two. Everyone is required to do pediatrics, obstetrics and gynecology, neurology, and psychiatry. And the ECMH at Northwestern replaces some of the primary care rotations that would normally be required. And primary care is just the doctors that you're familiar with, where you would go to your doctor for an annual checkup or for a regular exam. And then in the 4th year, there's some more rotations that would require some of the specialized knowledge. Some of the 3rd year rotations, or kind of tie everything together. That can be intensive care or an emergency medicine. And then there's some others where you can choose to do a sub-internship. So that's looking at a specialty in some of these fields. And that's especially relevant if you're interested in some of those subspecialties. And then there's also physical medicine and rehabilitation. So these are all the required clinical rotations or clerkships. And you can see that a lot of specialties are not represented here. So if med students don't know exactly what they want to do, there is quite a bit of wiggle room actually to fit in some extra rotations. And you basically have to fill your schedule. So you can choose to do some in some of specialties that not everyone is required to do. For instance, if you were interested in orthopedic surgery, you could, and didn't get to do that during your surgery rotation, you could do that. If you were interested in radiology or dermatology, you could also choose to spend some time with physicians that did radiology or dermatology and check those out. This is especially true of some of the smaller specialties, which don't necessarily have enough bandwidth to absorb all medical students from the class in every med school. There aren't enough otolaryngologists in the country to train all the med students for a few weeks in otolaryngology. So let's talk a little bit about some of the factors that people consider when trying to choose their specialty. Certainly money is important, and even people that are doing medicine for altruistic reasons still have to consider how much money they'll make, especially if they've taken on debt to pay for medical school. And you can see here that some of the highest paid physicians tend to be those that have a lot of training, especially surgeons. So I think in general, I don't have a graph showing this, but I would say that the longer you're training, the more that you are likely to make at the end of your career. But there's other things that are important. So there was a large poll conducted by Medscape where they interviewed more than 15,000 physicians, and they tried to determine who was the happiest outside of work. I don't know exactly how you phrase that question, and you'll see on the next slide that different specialties have different amounts of time that they spend outside of work. So you'll see that general surgeons tend to be quite happy outside of work. They also tend to make a lot of money, so that might correlate. But they also may tend to have less time outside of work, and so that's certainly something to consider. So yeah, you can take a look there, and it's a pretty interesting plot, and unfortunately shows that the happiest specialty, only 60% of doctors are happy, and the least happy in neurology, only 44% of doctors are happy, which is probably lower than it should be, and I think it's a significant issue in the field where doctors are burning out at pretty high rates. This is one last plot that I thought was pretty interesting, and it's an interesting plot to look at because it tells you about who's working how many hours. So you can see on the left, it has a bunch of different specialties listed. Some of them we talked about and some of them we haven't. And on the right, it shows how many hours with some distribution. The horizontal line the box represents the average in that specialty. The horizontal line represents a spread, the 95% confidence interval. And the vertical line at 0 represents how many hours a family practice physician works on average per year. So family practice is going to be, again, doctors that you would go to where they would see your parents, they would see you, they might even see children, and they would just do general checkups, and they may refer you to some of these specialists for some additional care if you need some more specialized doctor. I think in the United States medical system, a lot of people's healthcare is based on having a primary provider who's a family practice or general physician who would refer you to specialty care. You can see here the number in the column is hours above or below a family practice. And so that tells you how many hours per year each specialty is working more or less than a family practice physician. And obviously there's a huge variation here where some people will work more than this and some people will work less. But you can see that a vascular surgeon tend to have the most hours of any specialty. And they work 888 hours on average, more than a family practice physician per year. So if you're and that may not sound like a huge amount, but if you're considering becoming a vascular surgeon, that's 2.5 hours per day of extra work. And that's if you're working 365 days a year. So you can see some of the pediatric emergency medicine where they work shifts, work significantly less time, but it may be a little bit of a undercount because they tend to work 24-hour shifts or longer shifts. So they may have several shifts where they work for a long period and then several days off, entirely off. So that works for some people and doesn't. So this is not completely a black and white picture because you can see some work shifts and some don't. You can see why a lot of people want to be dermatologists as they are right at the top of the pay list and also right at the bottom of the hours worked list. But yeah, there's a lot of other things that people tend to account for. You can see that Celeste is talking about some blood cancers, and so she may be interested in certain specialties because of that. If you hear about my research in the future, you'll probably see that it's very based on imaging, and I may be interested in some of the radiologic specialties as a result of that. And so here, yeah, we're happy to take any questions that you guys have on this. This is just a flow chart which kind of plays into some of the stereotypes that people have about different medical specialties, and you can take a second to figure out, based on this totally non-scientific methodology, which medical specialty you should be doing if you are a medical student. I haven't done it for myself. I don't think I'm crazy yet, though, so I'm getting close, but I'm not there yet. It looks like I'm doing radiology. I don't know, we'll see. So yeah, that's all I have on medical specialties. I didn't want to go into every single one in huge detail because that's really, really boring, and hopefully this wasn't super boring. But I think all of us are happy to talk to you about specialties if you have any questions. So I'm pretty quiet on the chat today, though. What time is it now? It's 5, okay. All right, so no questions, it appears. I don't have much more than that. Next time, I don't know what we were going to talk about. Oh, there we go. Yeah. So I think that is a common conception. The question is about radiology with an MD PhD. Certainly, I think there's a few things. One is that MD PhDs really are flexible, and especially in the last decade or so, have been doing a lot broader of PhDs than historically so. I think most people, especially older people, tend to think of MD PhDs doing a hardcore biology wet lab PhD. And certainly there are a lot of MD PhDs that spend a lot of time working with animal models and cell models and doing biochemistry and biochemistry and biology. But there are a considerable number of MD PhDs these days who are doing a lot of other things. The NIH tends to fund science PhDs and not fund not science PhDs, but there are even MD PhDs who are doing PhDs in economics or doing PhDs in other fields that may seem less relevant to an MD PhD from this context. And even in our class, you can see that. I'm a biomedical engineer. I know I have done some wet lab work in the past, but it's been a very long time, and I would really struggle with that now. So that's not at all my expertise. And I think a lot more engineering and computer science oriented people are working in research with their MD PhD. So in my class, there are, I think, 12 or 13 of us, depending on how you count. And I think almost half of us are in biomedical engineering. So we have a really large number of engineers, and some of them are working in wet labs, and actually 3 of us are working in a radiology lab. So that's a pretty high, that's not a normal percentage for any class, but just in our class, 25% of us are working in a radiology associated lab. And in my lab, which is associated with radiology, we have 5 PhD students, and 4 of them are MD PhD students. So certainly MD PhDs have a really broad appeal. They tend to be relatively well funded as students, and they also tend to be quite hardworking because they want to get out of there. The training path is so long that it tends to be a relatively hardworking and quick PhD in most people's hope. So yeah, I think I wouldn't worry about choosing. If you want to do an MD PhD and you're interested in that, don't worry about the specialty. MD PhDs go into every specialty. Of course, there's some selection bias, and they tend to go into more medical specialties. but certainly anything is open to MD PhDs. Yeah, but if you're doing like neurosurgery, that's like way too much training. So it does happen. There are a lot of MD PhD neurosurgeons.
One of them runs the Mayo Clinic MSTP.
Yeah. How's work?
Great.
Looks great.
I can't see my own video, but I'm sitting here. I don't know. Can you see? There's mice in there.
Nice.
Yeah, nice.
So you can be hardworking like Sage if you join an MD PhD program. Yeah, I think that's a good question though, and I do think that MD PhDs are quite broad these days. If you guys have other questions, feel free to ask them. But I think there were some other things that we were going to talk about in the future. If you guys have any other input about other things you'd like to hear about, certainly we can give you guys more kind of, it seems like lectures are what works right now because we don't get so much interaction. But we can do some more kind of teaching on different aspects of physiology or medicine that we've learned about. We had talked a little bit about the heart and lungs and a little bit about COVID. So if there's any other things that you'd like to hear more about, we're more than happy to talk about those, or we can just choose ourselves based on what we like. You've heard about the other three's research. You have not heard about mine, and so I can talk about that at some point if people are interested. And then I was going to give a brief talk about sort of general recommendations about medical school and applying to medical school. I know there was some interest in that, so we could do that shortly too if people are still interested in that. Any feedback? Looks like it gets to be our choice. What are we going to talk about? Okay, more about how to apply to med school and stuff. I don't know about the and stuff, but certainly we can. Yeah, certainly I think we can talk about applying to med school. I know that's a stressful thing, and there's a lot of requirements and things that people have some disagreement about doing. So we've talked a little bit about that in the prior chats, but I think we can certainly do a quick presentation on that. And again, it's very clearly going to be our opinions and not any absolute facts, but we've all done it, and a lot of us have participated in the admissions at Northwestern and have some not insider information, but some intuition about what makes a strong applicant.
And I think we can also extend that conversation to like college, although we talked about that a bit before too.
Yeah, certainly.
Closer to your timeline also.
Yeah.
Because how many people are seniors next year? I'm assuming that none of you guys are seniors now, right?
Actually, I have a poll for that.
Oh,
Yeah.
let's do a poll.
Let me see if okay, not that one.
Okay, we have 1, 2, 3, 4, 5, 6.
But what does me
6.
mean?
I think it means senior next year. Oh my
Senior.
God, look at that, Blanca.
I know Blanca's got pretty tech savvy.
Yeah. Wow. Quick
Seems like we're
response.
heavy on the seniors. Okay.
Rising seniors.
Rising seniors.
Yeah, rising. What grade are you in? 11th. Yeah. Well, come on. I don't think seniors right now are listening to us ramble on. I think they're finishing.
Well, that's what I figure too.
Okay. We may have some that are in 8th or younger as well, but yeah, okay. Are you guys interested in talking about college stuff? Because med school is a little further off for you guys. We can definitely talk about college too, though a lot of the requirements are changing due to COVID and because we're old, but we can certainly talk about it. Okay.
And
Oh,
the
cool.
poll, I think a few people just didn't feel like answering today. Okay, There we go.
No, I
So
think we got some in the chat as well, but I think we're pretty good on the poll. Thanks, Blanca.
no problem.
Okay, cool. So that sounds good.
What kind of questions do you guys have about college? Like,
Which schools have good research and biomedical engineering programs?
Yeah.
You can probably find that online.
Yeah, so a
Schools
lot of
are-
the questions about like
They're
which,
ranked pretty tightly, or pretty like schools are tracked very closely.
yeah,
And-
there's a
and
lot of rankings online. I think generally good schools have good programs in most things. What happens if you switch majors? Yeah, that's a good question. Yeah, maybe we can talk about college and med school next time or something like that. I'm happy to talk about switching majors now, but certainly we could have an entire session on that.
yeah, and if you guys have like questions that you can think of now, like, yeah, it would be great if you could put them in the chat so we know like what to put in the presentation next time to make it as interesting as possible through Zoom.
Yeah, we'd definitely like to answer your questions. So if you guys are comfortable talking to us, talk, but I don't think that's going to happen. So enter some messages in the chat and we can prepare some type of presentation. If you guys give us questions now or send them to Blanca, we'll try to answer them ahead of time. And if you don't think of them or you think of them while we're talking, definitely we'll try to answer them on the spot. But yeah, there's going to be a lot of questions about like which school has this or does the school have this that's good. I know for a fact that all of you guys are experts in using Google. So some of those questions are going to be better answered. Like if it's a very specific question about like, does the Ohio State University have a good bioengineering program? That's like go to US News and World Report and search for their ranking. So there are a lot of other factors outside of the ranking which are quite important as well. Yeah, so I think we can talk about that when we talk about this, what things we should do during med school, during undergrad to prepare for med school. Yeah, there's certainly a pretty concise list of things that are like pseudo required. So they're either stated as required by med schools or they're kind of just expected, even if they're not explicitly required. So we can talk about that next time.
What
The
makes
ranking,
up
that's
the ranking?
Money.
rankings are very contentious and
Money, and a lot of the ranking is also dependent on how many people apply versus how many people get in to a given program.
Yeah, but we can talk about rankings. We can talk about that. That's a good question. Okay. Most questions, if you look hard enough, the answer is money. But unfortunately,
that one you don't have to look that hard.
MCAS. I was on MCAS today. So
Really?
no,
Why?
I was on AAMC. Sorry, I was looking at specialties. So there's like you guys can join some of these organizations that are kind of free-ish, or you can just Google it and find things like this. But there are some like calculators, and I'm sure they have them for undergrad too, but I don't know if they had them when I was an undergraduate, but they may have them now, where they ask you a series of questions, and then they give you so they poll people that are leaving colleges and med schools, and they say like, what did you study? Or what is your specialty going to be? Or what residency are you going to? They ask them kind of outcome questions. And then they ask them a whole bunch of questions about like their personality and what they enjoy and that kind of stuff. And so then they can make a test, like a quiz, basically. It's kind of like BuzzFeed for your career. And you can answer a whole bunch, like sometimes they have a ridiculous number of questions, like 50 or 100. But you answer the questions and then it kind of gives you a breakdown of like which careers and specialties would be most compatible with your personality or what you kind of envision your future career to look like. So I know when we came to medical school, at least in the class that Celeste and I are in, they kind of made us do one of these quizzes about like which specialty should you do based on your like personality and stuff. So it's actually kind of fun. You can see what other people that have a similar personality to you might enjoy, and this can give you some good ideas. Obviously, that should not be the only decider in what your college major is or what your medical specialty is, but they can be fun to do.
Do you remember what you got?
Do I remember what I got? I remember what I didn't get. Very low chance of me being a psychiatrist. I think I had general surgery and orthopedic. I think I was a real bro. Yeah, I think I had
Yeah.
ortho and general surgery.
Should we all take it and we can put
Yeah,
it
maybe
in
we can take it again. I don't think you can take it again, but I think you can pull up your results. I'll figure it out. We can see.
Yeah, I haven't logged in on there in a long time.
Yeah, I had to change my password. It took like a long time. It's
for
challenging.
AAMC or AMCAS?
Whatever. I
Are
don't
we
know.
still
There's
on?
too many-
Are we
there's too
still on
many-
a tangent?
There's too many acronyms. Sorry. Yeah, we can talk about using MCAS as well. I'll have to remember which one it is, but I can definitely figure that out by next time.
The other three of us can talk about it.
That's true. You guys could talk too. I've been doing
Steven
way.
can talk.
No, I don't like that one.
Yeah, you haven't been doing so much talking, Steven.
I've been quieter.
And
Okay.
normally you're such a little chatterbox.
Yeah, I think I'm call brain.
Oh yeah.
Oh yeah, when are you presenting?
I did, so I am representing soon.
Okay.
Representing.
All right. Well, thanks for the questions, guys. I don't think you guys need to sit around and watch us chatting. If you have any other questions, we're happy to answer them. Otherwise, next week, maybe we'll talk a little bit more about college admissions and med school admissions. They're pretty similar, just different requirements. I don't know if you have anything else, Blanca.
Nope, I think that's all for this week. If anyone has any questions, just email me and I'll forward it to our volunteers. But thank you guys. We'll see you next week.